make sense of it all for you, I'm going to talk about why it's hard and some things that we're trying to do. Just to correct the record, I have an appointment in computer science here, but my home is down at the medical school in Denver. So I'm going to speak up for Denver for just a second. All right? Oh, come on. It's a nice place. All right. Can we do the slideshow? And is there a clicker or something? Here, take this guy. Great. So I feel like the Monty Python here, because this is going to be completely different than everything else you've heard so far. Because I want to talk about some of the how many computer scientists does it take to start up a slideshow? We have this conversation in my lab all the time. All right, good. So there are some big gaps in this world, the gaps between the basic scientific understanding of human health and disease and basic scientific understanding, then human health and disease. And I'm going to try and talk about jumping over some of those chasms. I tried to find a good chasm shot from Colorado. This is New Zealand. I apologize. I'm going to have to, all right. Takes another computer science stuff. It's beeping at me and not doing anything. Boy, that's sad. All right. Well, so this is a quote from the current issue of Surgical Oncology, and I just love this quote. And this is a little bit in contrast, perhaps, to Kevin's talk in the beginning, although I have a slide that he used too. And it's this kind of retrospective on how a decade of genomic information has changed the treatment of cancer. And you can almost hear his bewilderment at the end of this, okay? God, it's so much more complicated and heterogeneous than we imagined. We were looking for an individual biomarker. Oh, God, I'm going to have to click next on each one of these. All right. So, and they just turned out to be hard to find. And they're hard to find for a couple of reasons. And one reason they're hard to find is the haystack is so big. And the graph, maybe at least the pointer will work. Yes, this graph, notice that this is a log plot. So for those of you who aren't scientists, this is about 1 million and this is 1 trillion up here. So it goes up very fast. And this is the amount of sequence information in GenBank. And it's been going up in this kind of exponential way. It may be flattening out a little bit, but it's been going up like crazy. We've been sequencing a lot. You heard about thousands of human genomes. There's an effort that Hausler is running to go look at tens of thousands of animal genomes. And it's been fascinating and a lovely scientific ride. We've learned a tremendous amount. And most of what we've learned is it's more complicated than we thought. And that's kind of a sad moment for us because we have some really pressing issues. Larry, in his opening slide, mentioned diabetes and cancer, and I'd like to add mental illness to that, which are complex systems diseases. They are not the fault of a single gene or a single gene product or a single defect that you can go in and with surgery or with a particular drug go after. They're much more complicated than that. They involve hundreds or thousands of gene products and their interactions. And that's the thing I want to focus on in this 40 minutes or so, is to talk about how we're going to unravel those hundreds or thousands of interactions. It's a very big task. And I also want to point out, and this is, I stole a little bit of thunder from Jamie Haywood, who will come later, that we're getting much better at characterizing people. We're terrible at trying to figure out the genetics of what. So we think of diabetes or heart disease or cancer as a unitary disease. There's one number that goes in the diagnosis slot in the patient record that says, okay, you have diabetes. But that's crazy. People have tremendously different kinds of diabetes, different stages, different kinds of courses. The phenotype that we characterize is really not up to the detail that we characterize genotypes in at all. So we have to do a much better job. And fortunately, computers are helping us there too, of differentiating between different kinds of patients and their individual histories and their individual outcomes. And it's there that we have to connect up those thousands of gene products to those very different human stories. And that's a critical piece. And we're finally getting some information about that. Phenotyping, the description of human beings, is getting better, not just because of these social networks that give us detailed daily information about how am I doing with my disease and what did I eat and how much do I weigh and all that kind of stuff, which is really useful. We also have these patient records, which sadly, now that I've looked at them in some detail, are much less useful than the stuff on social networks. This is real patient data from real insurance companies, which are the ones who actually possess almost all the patient data. Okay, hospitals and doctors don't aggregate it; they don't have it together, and the sort of regional health information networks that are supposed to put it together can't. And so what we get things like are how many days since the first stay. Okay, that's useful for payers. They want to know that kind of stuff. It's helpful to figure out how much you're going to cost, but it's absolutely worthless for describing your disease. So we're going to have to do a better job of the phenotyping to figure out all those details. All right. So a lot of this is, I have, every time I give a talk, I have to say something that's supposed to rile up Larry. And so this is my slide that's supposed to rile up Larry. So he's busy out there looking for biomarkers. He's going to find a marker that will tell you whether your lung cancer is going to get better or not, or what drug you ought to take. And furthermore, all the pharmaceutical company guys here are looking for magic bullets, okay? We want a very, what did you call it, a clean drug? It was a lovely, lovely word. It's something that has a single effect. It only affects the target and nothing else, okay? And I think both of those ideas are, they're not quite the ash heap of history. They've been useful in certain ways, but are not the future at all. And this should tell you why. This is the result of more than 500 genome-wide association studies. Each one of those dots is a disease gene, okay, that took usually millions of dollars to find. And along this axis is the odds ratio. If you have the bad allele, what are your chances of getting the disease? And notice almost all of them are down here along the bottom. Okay, that means you might have a 20% elevated risk of getting the disease, a 40% elevated risk of getting the disease. If you have the gene, you are not a dead duck. You don't have to worry about it too much. And up here, this is the reported risk allele frequency, okay? So these are the people who have the disease. How many of them have the bad allele? And it's not completely around zero here, but a lot of them are pretty close to this. Less than half the people who have the disease have that risk allele. So we're not really understanding one gene at a time what's causing these complex diseases. Rich Spritz, who was a speaker last year, used to like to say in his Human Medical Genetics seminar that the only normally inherited Mendelian gene in human beings is red hair. That's this one right here. And it is, if you have the MC1R, the right allele, you have a 15-fold or so increased risk of red hair. And that's almost Mendelian, but notice the penetration is very low. Only about 10% of the people with red hair have that gene. There's lots of different ways of getting red hair. And that's true for almost all of our diseases as well. All right. I hate having to walk over. There's a reason for this. Okay, we understand this, and in fact, we've understood it for a long time, and that's because human beings are extremely, and other organisms, all of us, are extremely complicated networks of interactions. We're not just one gene does one thing. Every time you read in the newspaper, scientists discovered the gene for X, whatever X might be, or you hear your colleagues say that, they're lying. There is no the gene for anything. Okay, there's hundreds of genes that work together in concert to make it happen. And the way they do that is through these complicated networks. So this gene turns on that one, which turns on this one, which interacts with that one. And if you have a drug and you shut off this bad guy up here, you got that cardiovascular drug, the network just correspondingly changes. We change the network. These are homeostatic networks. It's a key word in the history of biology. It means the network wants to stay the same. It's trying to keep you alive. All right? And so if you come in and change it, you have a magic bullet, and you take out that one gene that you think is the bad guy, the network will adapt and figure out how to live without it for the most part. And that's true it's a very important fact to remember. And it's kind of why some of the drugs that we've been using to go after these systems diseases like cancer and heart disease and diabetes haven't worked so well. There's a lovely picture here from a Hornberg article from a few years ago. There's a very common sort of thing that happens in these networks where you get some kind of signal, and that hits some sort of receptor, and then there's a signal transduction pathway that involves some feedback to amplify that little signal, and then something happens. This is proliferation, so it's cancer. And so you aim a drug at this broken gene in here. It's this signal transduction gene that's broken, and if we could inhibit it or turn it on louder or something like that, we'd cure the problem. And then we come up with a drug that does that, and it barely does anything. Okay, it hits the target, great. But people don't live. We see that all the time, right? We've got failures in the cardiovascular drugs. The CTEP story from the earlier talk fits. So maybe the thing to do, because this is in the middle of this feedback loop, if you affect this drug, all it does is make more of the receptor, so the broken thing doesn't have to work as well. And so maybe we should be targeting things that aren't the broken gene, that are some gene that isn't broken but is upstream of the broken one or downstream of the broken one. And if we did that, then we'd get, because we're outside of the feedback loop, we'd get a response to the condition that looked more like the response of the target. And so this is, I think, one of the fundamental reasons that the magic bullet and the biomarker stuff hasn't worked, because we haven't understood the nature of the networks and the way that networks work together. So how are we ever going to do that? We gotta figure this out somehow. Unfortunately, the path we've taken is we'll find the network. Some of you are old enough to remember the Manager Borheim wall chart that had the network on it. Okay, it was the whole metabolic network, and we all had it up on our walls, and we'd look at it and think, God, it's complicated. It turns out that's, first of all, a small fraction of the real signaling networks and metabolic networks in humanity. And furthermore, there isn't a network. The network changes. It's dynamic. And in fact, one of my favorite papers of the last year, it came out in Science in December of last year, and it showed that the networks completely change in response to DNA damage, which is, of course, what we do to people who have cancer. We give them DNA damaging agents. And so their entire set of networks all shifts out from under you. So if you thought you were targeting the signal transduction of the broken thing that was stuck on, one of the reasons those drugs doesn't work is we give it to you in the chemotherapy context, and that means the whole network is rewired. And so we can't make easy sense of just changing one little piece or attacking one little piece of the network. The bottom line is these systems diseases need to be hit with systems therapies. We need drugs that aren't one-target magic bullets, but that have a whole range of effects that affect that whole network in ways that help. For cancer, at least, you could think about synthetic lethality. That's an important idea from biochemistry, where if you knock out one gene, nothing much happens, and if you knock out another gene, nothing much happens. But if you knock them both out together, the organism dies. Okay, that's synthetic lethality. So what we'd really like are synthetic lethal systems perturbations. So cancer is a systems perturbation. It's changed that whole system of the cell. And what we want is a drug that changes the whole system in such a way that the combination is lethal. And if you don't have cancer, that drug won't change your cells in a way that's lethal, but cancer cells will die. That's synthetic lethality at a network scale. So that's what we're really looking for. How are we going to get there? Sorry, Kevin already did this. I don't have a pretty picture of Bruce Korf, but I think he was right on. Okay, all this genomic sequencing is wonderful. And it's not just about genomes, by the way. There was a question earlier that said, well, can we look at the epigenome? Can we see about stuff? And the sequencing technology is really, it's almost magical. It allows us to look at a lot of phenomena inside living organisms in ways that are really illuminating. And there are a bunch of very clever people building methods and the like for doing things like looking at nucleosome occupancy and RNA and the like. And furthermore, one of the things that really surprised us relatively early on is it looks like the whole genome is basically transcribed, right? Only a tiny percentage of it is protein coding, but all the stuff that isn't protein coding gets made into RNA anyway. And we've discovered all sorts of interesting things about that. Some of those RNAs actually do some work. How about that? And so we have a lot to do in understanding this stuff, and we're just at the beginning. 10 years is not very far into it, and we're not at $1,000 genomes yet. And I think Russ Altman, who has he's an MD, PhD biochemist with a strong computational piece, he's kind of the renaissance man of our field, pointed out that not only is sequencing cheap, but computers are cheap. What's hard is understanding. And so now I want to shift gears a little bit and talk about how we're going to get to understanding. One of the things that we have is a tremendous collection of databases of information, gene-centric information out there. And so you can't read them very well, but these are screenshots from a bunch of different databases. And it turns out academics like to get publications and they like to get credit for it, and the people who ran the databases didn't get much credit. So Nucleic Acids Research did an amazing service. And so they annually publish a journal issue where you could submit your database for peer review. And if it passes, you get into the database issue and you can get some publication credit for all the work you did to make that database. And that was great. The first issue had about 100 databases in it. Last year, the 10th time, 11th time they've done this January, there were 1,300 databases in this thing. Okay, some of those have millions of entries in them. There's almost 900,000 peer-reviewed biomedical journal articles published last year. We know an awful lot, but we can't put it together in this systems way very easily. And I want to take a quote from one of my least favorite scientists in all of history, but who said it beautifully, which is that two paradoxes may be better than one. If we have too much information, if Mae West is right and too much of a good thing can be just wonderful, how are we going to make it wonderful? I'm going to suggest computers are the answer. So we already have this problem. You heard it maybe in Susan's talk, where she gave you that laundry list of genes. There were kind of 50 some odd, 100 genes that played a role in placental and preeclampsia stuff. And The funny name, that pigmentation name that came from eye disease, this is not unique to her, okay? The problem is genes have been studied intensively in narrow areas, and they don't believe in disciplinary boundaries. Genes will do stuff all over the body, and they won't pay attention to the fact that they were first studied in the eye and got named after eye and stuff like that. They're going to do whatever they do. And so we have this real problem that scientists have worked by studying narrowly, intensively down in one area. They know about all the genes that play a role in that area, but they've never heard about the genes that play a role in some completely different area. How are they going to come up to speed if there's 50 or 100 genes that showed up in their experiment as relevant to whatever I'm studying, but have been published on in 50 different fields with hundreds of papers? How am I going to come up to speed on that stuff? My hope is that computers can help. This is me, by the way, getting creamed by Watson at Jeopardy. I want to suggest that we haven't actually gotten to artificial intelligence yet, but there have been some important contributions. The first one has really made a big difference in molecular biology, and this is a place where Larry and I used to disagree, and now he's come around and seen the truth. Okay? This is an old-fashioned kind of AI. We've been doing this stuff for, 20, 30 years. And the idea is we want to distinguish between the red dots and the green dots. And we have some yellow dot, we don't know if it's red or green, and we want to guess. And statisticians have been working on this for 100 years. And statisticians said, well, you draw a line, okay? And the ones above the line are red, and the ones below the line are green. And if there's some on the other side, well, we call it noise, and we don't understand it, and we write it off. Then, about in the 80s or so, we came up with this stuff they call artificial neural networks. And it was inspired by neurons, although it doesn't really work very much like them. And it could learn any function at all. And so you learn functions that very carefully distinguish the green ones from the red ones, but they did something we call overfitting. That meant they were so close to the data that they didn't generalize very well, and they would make these mistakes about seeing only exactly what they had seen before as the green ones here. And so this is overfitting. And what we'd really like to do is draw this actual circle here. This is all metaphorical, but it works in higher dimensions. Don't worry about that. It just means more variables. So what we've come up with in some of the machine learning stuff these days says, well, I can't draw some line, and I don't want to draw just any line. I'd like sort of a regular family of curves I can use here, but I can't define them ahead of time. We can do quadratic and things like that, but I don't want to have to specify them. So what the machine learning stuff does is take a function that takes any two of those points and says, how similar are they? Are these two really similar to each other, or are they kind of different from each other? And then uses that to project the points up into this high-dimensional space where we can use the statistical trick of drawing straight lines. And this works really well. It allows us to integrate multiple different sources of information that we don't know how to integrate in our heads, and just say, okay, all you have to do is put them together and tell me, are two things close or far away, and I can start making good predictions. And this turns out to be useful even in the biomarker world. How about that? There's other things that have come from AI over the years. I'm not going to go into any detail on these things, but they've actually been most useful in trying to understand what's going on in sequences. So we'd look at some kind of probabilistic models that say, what's the chance of going from a T to a G or something like that, and we can train these guys to try and figure out, for example, these are all splice sites. And then we can get further, more complicated, and we can use grammars and say it's not really whether it's a T or a G, but it's whether this T lines up with that G over there. And so that's a grammar will allow us to do that. Turns out to be very important in RNAs. And all I really want to point out from this is there's been a sort of steady drumbeat of information or methods from artificial intelligence that have kind of crept over into biology that have turned out to be useful in a small way. And now what we want to do is make it useful in a big way. We want to take those thousands of databases and millions of articles to help us understand what's going on in those hundreds of genes and their interactions. That's really the critical thing. So we went and have actually been following around molecular biologists using computer tools trying to analyze data like this and try to figure out what are their information gathering needs. What do they want to know when they're doing this stuff? And it turns out they're fairly straightforward in some ways. They have this question about, well, what's the explanation? What's a possible story that I could tell that would link together all of these observations that I'm seeing? And these explanations are a really critical feature of humanity as well. So they were studied by a philosopher named Charles Sanders Peirce, who came up with the unfortunate name of abductive inference. It really has nothing to do with aliens or kidnapping or anything like that. What it really is about is, what's a plausible explanation? It doesn't have to be the correct one, but what would hook these things up together? And biologists have lots of versions of these things. Why is the structure present? Why is this function being realized now or here or something like that? And it turns out, any of you who have teenage children will know that you can't just ask why. You have to provide a set of alternatives to choose from. Why didn't you do your homework? I got an endless litany. What I really want to know is why you didn't do your homework and you played video games instead. Okay, that's the question I want the answer to. And so that's contrastive abduction. And so the problem with real abduction is there's an endless sequence of causal factors that led to whatever happened. Okay, why did I have cereal for breakfast this morning? Well, if the state of the universe at 10 to the minus 10 seconds after the Big Bang had been different, I would not have had cereal for breakfast this morning. That's part of the explanation. Okay, it's not a very interesting part of the explanation. So what I'd really like to say is, why did I have cereal instead of eggs? That's the question I want to have I can answer. Okay, so that contrastive abduction stuff is something that computers can go after. And at least we can find examples of it in people's reasoning with computers. It's all over papers. So if I go look in papers, and this was taken by Elizabeth White, who's doing a project on trying to find explanation in text in biomedical journal articles, you find it constantly. The language of biomedical journal publication talks to us about possible explanations. We call them hypotheses often rather than explanations. Scientists are loath to say cause, but nevertheless, it's the same phenomena that we're going after. So, when we try to help scientists, we try to build software, help scientists understand this stuff, we often give them lots of possible explanations or lots of information that should help them build those explanations. And, unfortunately, it often, you can't see these too well. It looks like this. You couldn't see it too well in detail on the screen either. So this is about 150 genes that play a role in response to beta blocker therapy and heart failure. And so that's an important thing because it takes physicians 6 months to figure out whether beta blockers are helping you or not. And if they're not, you needed to be on a transplant list 6 months ago. So it's a really important kind of thing. We'd like to find some markers that would help us understand both why it works or doesn't work, and at least predict ahead of time. And so we put up something like this on the screen, and the biologists say, okay, show me fewer. I cannot figure anything out from this. I'm not going to go click around on all of these things, which is what we ask them to do. So there's too much to absorb. Not only do we have to provide explanatory material, we have to help people prioritize what pieces to look at first, what sort of thing would be most interesting to them. And so computer scientists in the visualization world have tried to write these degree of interestingness functions. So I'm going to rank every one of these genes and say, okay, which one is the most interesting one? And that's a great question. It's really a fundamental to human intelligence, much the way explanation is, because one of the things that we care about, there's no one thing, but one of them is, well, how certain are you about this? It would be more interesting if you gave me something I could believe in rather than some hoo-ha that somebody made up and published in some God knows where. So that matters, but not exclusively, because sometimes I'd be interested in things that are very uncertain but would be really powerful explanations if it turned out to be true. So connecting with the things that the user is trying to do. What was the point of that study? Why did I do that experiment? Well, I'm interested in some particular diagnosis, or I want to test some particular therapy, or the like. And so that helps pick out which are the interesting ones in there, which ones connect up to the things that I'm interested in. And also, each user is different. Everybody who looks at the data looks at it with their own perspective and their own interests, and the papers they've read, their bibliography, the papers they've published, perhaps, what's current, what's been published on recently that I might not have read yet. All of these are different ways we can try and pick out pieces of this puzzle to present to people. And so we try to come up with programs that can do both of these things, that can hypothesize interesting explanations. The task here is I got 150 genes that came out of my experiment that play a role in the phenomena I'm studying. What am I supposed to do with 150 genes? If I go and print out all the articles that have ever been published on those 150 genes, I get a stack this high. And mostly people won't read it. They need some more efficient way of understanding what's in there that's relevant to them. Because mostly I'm a cardiologist, I don't want to read the oncology literature. I really don't. And so I need some computer program that can take this set of genes and run it through the literature and give me some story that can help me understand what I'm seeing. And so we have systems that do this already. I'm not going to talk in very much detail about how we do it or what it is, but there's a lovely YouTube video. If you search Analyzer, and I'll show you how to spell that in a second, you'll see people at work doing this. But it takes 3 different pieces. We call it the 3 R's. One is reading. We have to write computer programs that can take information out of those databases, and most importantly, out of the literature, and put it together into a representation of what it is that human beings know about molecular biology, and integrate all of those disparate sources of knowledge that don't all agree with each other, and weren't all done in the same context, and don't all fit neatly together, but put them together so that we can take everything that the program can represent about what's out there in the literature and align it with the data that came out of your experiment. Because we really do have to swallow the information about the 150 genes. Got to do it. And this is not like Google, okay? This is not trying to find a needle in a haystack where there's one piece of information. If only I knew that piece of information, I would be happy. The whole haystack is made out of needles. They're all potentially interesting. I need to understand them all. And so you kind of got to swallow the whole haystack full of needles. It's a very miserable metaphor, but it's what we have to do. So part of the trick is not just aligning it with the data so the computer knows what's relevant, but we have to present it to people. So an important piece of what we do is the visualization. The third R is reporting. We want to put together visualizations that interact with people so they can explore stuff. We'd like to automatically summarize things, produce texts as well. And so those are the 3 R's of building an AI system that I think can really help us take apart this overwhelming amount of information coming from our post-genomic experiments and make sense out of it. Analyzer is the system we have now that does this work, and I encourage you to go watch our YouTube video. Analyzer does not come up with explanations on its own. Analyzer is a tool to try and help people come up with explanations. And so it's a way of trying to help biologists align all this knowledge from all these different sources with the results of their own experiments. And it's had some pretty impressive, I think, early applications. One of them was in understanding craniofacial development. So if you go back and you look in PubMed and read the Analyzer paper, you'll see we're trying to understand a bunch of complex genetic interactions in the development of the mammal face. Recently, we had this really cool results in analyzing this heart failure data that I mentioned to you before. And I think it's a good example of why explanation is different and important compared to what we previously have done. So the statisticians went over the same data that we went over and came up with this biomarker. Okay, we filed for that, by the way. That's a good biomarker for trying to predict responsiveness to beta blocker therapy. And all they could say was, it's the single best predictor we have of outcome. It's got an R-square of 0.4 something or other, which isn't spectacular, but it's good enough to be important. And what we could do is say, well, gee, we're going to look at the context of this thing and see, gee, what does it do? What are its downstream effects? This is a basic helix loop helix. It's a transcription factor. What are the downstream effects? That's kind of interesting. But even more interesting were what were the upstream things that turned it on. And we came up with a story around hypoxia, which got all the cardiologists really excited. Oh, that makes sense. I understand why hypoxia signaling would play a role in this. And by the way, it connects up right to the receptor that the drug targets. So that's another good sign. And so we could put together a story that you couldn't put together with just statistics. And you couldn't put together with just the genes that the expert happened to know about. We had to take all this information from all these different pieces and put it together in a comprehensible way, so that the people who cared, the people who designed the experiment, could make sense of this stuff. And that's really the goal. That's the hard part. So now I want to make, Larry said be outrageous, so I have no problem with that. I'm going to make a very strong claim. Oh, you like this? Good. A very strong claim. And that claim is, we will get to artificial intelligence, okay? I don't think Watson is. Watson's a very clever game-playing system. Deep Blue, very clever game-playing system with logic theorem prover. You know, it turns out that the history of artificial intelligence is the history of doing things that kind of white guy computer scientists think are cool, right? Proof theorems, play chess, be good at Jeopardy. We'll actually, one day we'll get beyond that and do some more interesting kinds of thinking. And Seymour Papert had a wonderful line about this. He said, and I think it was earlier than '74, but that's the first I could find. And you can't actually read this, you have to hear it. You can't think about thinking without thinking about something. So what something should we be thinking about if we're writing programs? Okay, and I like molecular biology because the real holdup in artificial intelligence has been what's called common sense. Okay, the things that 5-year-olds know about the physical world and that 15-year-olds know about the social world, okay? It turns out it's enormous. They know a lot. And nobody's written it all down. There's no textbook that has all the things that 5-year-olds know about the world in it. Okay, and if they did, it would be a huge textbook, and it would be hard to represent and all that kind of stuff. So molecular biology is nice because there's no common sense in it at all. Okay, there's nothing you learned as a 5-year-old or a 15-year-old that matters in molecular biology. Everything you know about it, you got out of a textbook or a journal article, or maybe a few of you out of an experiment in your own labs. But by and large, that large amount of information is explicit. We could figure out how to get it into a computer if we worked at it, right? And furthermore, it's everything. I love Huxley, and this is a great quote of his, the study of human physiology, by which he meant systems biology. Systems biology and physiology are the same thing. There is no more comprehensive education than that. Okay, it calls into play every branch of human knowledge. So if we could do this, then we can do it all. We will have artificial intelligence. So are we going to? Larry mentioned in his opening line that there were these other omes, and I think he said EEGs might be interesting. We'll do a brainome. Well, it turns out there is a brainome. It was published 2 years ago, and it's a white matter fiber tracts. So you can use DTI, magnetic resonance imaging, to try and figure out what all the connections in the brain are. And it's a really beautiful, there's stuff at UCLA that I stole this from, that sort of shows all these connections, and you could try and look at them, and there's really beautiful information in there. But I'm going to make a very high-level, gross generalization about what we found. That's only sort of true, but it fits, I think, nicely with the AI story. And that is mostly the human brain is very densely locally connected. We have pieces of brain that do specialized things. They recognize the cry of a baby that's hungry versus one that's hurt. They recognize hands and them reaching for the gun or whatever else it is in there. Okay, there's all these very finely detailed local calculations. It's kind of like the machine learning stuff and the HMMs and all the other things I showed you that were AI techniques that apply in molecular biology. There are relatively few pieces of the brain that are really widely connected to all the other pieces of the brain. And if you squint at it right, there are 3 of them. One of them is the uniquely human bit, that huge prefrontal cortex that we have up here. And I like to think of that, some people call it executive function, but I like to think of that as that's where the explanations are. That's where we integrate all the pieces of information, because explanation can draw on anything. There is nothing you know that couldn't be used in an explanation of something, somewhere. And it's not only finding the path between the thing explained and the thing that explains it, it's how well that path fits with all the other explanations we have in our head. Because our head is full of explanations for everything. If you've been around a toddler who says why all the time, they really want to know, and they remember and they keep it. If you give them nonsense answers, and there was a horrible psychological study that actually looked at this and gave little kids nonsense answers to their why questions, they stop asking them. They don't just want to hear you talk. They really want to know why. It's important. So I think that's what that prefrontal cortex piece does. So explanation is a central role. The next piece that's connected up is a very ancient piece of the brain called the limbic system, which is where emotion is, it's typically said it's where emotion lives. But I actually think it's not quite emotion. It's our desire. It's the things that reward us, the things we want and fear. It's all those wannas and don't wannas in there. And to me, I think in the metaphor here, that's our interestingness function. That's trying to figure out the value of everything. And we have to do that. Everything that we're confronted with, evolution says, better figure out whether that's valuable to you or not, because if it's not, ignore it. And if it is, pay attention to it. And that's interestingness, at least for us. And then the last piece, which was a big surprise, because I think it's not too surprising to a lot of neuroscientists that prefrontal cortex and limbic system are connected to everything. This is cerebellum is connected to everything. Cerebellum, that funny little thing in the back that we used to think was really just sort of posture and gait and stuff like that. It turns out cerebellum is connected to everything. And my guess is just that it's all about how human beings and other creatures understand sequence and pattern. It's about timing. And if you think about it from the perspective of a musician, that's really where the soul is. It's all the details of the specifics of the timing. So I'm not sure we'll get computer programs to do that very well yet. But the other 2, I think we're on the way to. Why would you care? What is the payoff to real people who are sick? You know, you wanted me to talk about health, and I don't really have too much to say about it at the moment. I think the answer to that is the payoff of computer systems that can do a better job of explaining genome-scale data and finding interesting bits of it to draw our attention to is not going to be immediate for anybody who's sick now. The best I can offer is we may have a payoff in changed clinical decision algorithms for heart failure patients. I think that will come fairly soon. Mostly it's a long, slow slog to get some basic understanding that you can find in a laboratory somewhere to do something that you really want to change the way millions of sick people are treated. And we've got to be patient and recognize that. It's hard to get there. And especially if I was right in the beginning. And the answer is we need these systems drugs that are going to have lots of effects, not some single target, but are going to perturb the whole system in ways. Or we're going to do combinations of drugs, which I'll get to in a second, which is sort of the simple-minded way to try and get to that. It's going to take a long time before we figure out how to regulate those things, let alone figure out how to really invent them right. We are early days in this stuff, and we need not to oversell. Taken from an AI guy, okay? Don't oversell. All right. Fundamental bottom line, other than be patient, systems diseases need combined therapies, okay? If there's a network problem, and it's not just one broken piece of the network, it's a whole network that needs to be dealt with, then we need to deal with the whole network. Combinatorial therapies, where you give drugs that are supposed to hit different places in the network simultaneously, are already a little bit important. So we do that, it's the standard of care in HIV, so we do it clearly in HIV. It's starting to happen in a few cancers. There was a lovely article day before yesterday about treating pancreatic cancer with a 4-drug combination that really seems to make a big difference. And so I think we're going to see a lot more of that kind of combinatorial therapy where we know the different pieces of the network. We can see if I push on here, it compensates over there, so I'm going to push on here too and find enough places to put our whole fingers in the dike that it actually works. And that's a tough task because combinatorics lead to combinatoric explosion. It's a fundamental idea from computer science. Okay? The number of possible combinations goes up very fast. So, for example, there was a lovely paper from earlier this year, I think, where we're screening for combinations of drugs, and this was in the NCI cancer line. And so there's about 600 approved drugs. You take all 600 and combine them with all 600 others. That's 360,000 pairs. Okay, that's a small starting number compared to the number of genes. Think about the number, there are 20,000 genes. How many pairs of genes are there? That's your homework for tonight. There's a huge number. They screened all 360,000 pairs and found one interaction on an antifungal drug and an antibacterial drug that seems to slow down the rate of progress in lung cancer. Who knew? Okay, so the combinatorics are going to be where we have to go, and we can't go there on our own. It is too big for the human mind. Hopefully, if Russ Altman was right and computers are essentially free and the sequence data is essentially free, then we will be able to go after this stuff even though it's too big for us to think about on our own. We hope to get to the point where we have programs that generate interesting hypotheses from phenotype data, from genomic data, from all of that, and will be our genuine partners in this process of trying to get to real healthcare. I think that's all I have to say. I would be glad to take questions. I want to acknowledge the wonderful people in my lab who did most of the work here. The money that by and large came from NIH, not enough of it. The government is really under assault in this, and there are philanthropists here who want to talk. I'd love to. And most importantly to Larry, who has made such interesting, stimulating explanations and kept us all on the edge of our seats. Thank you very much.
Wanted to thank you for both a very stimulating intellectually and entertaining talk and comment. Well, I first have to say I love the notion that systems biology is the good old word physiology, and we don't need to invent a lot of new ones.
New ones are useful.
But the comment with slight disagreement on your notion that combinatorial therapy is not the standard. If you actually look from a patient-centric view, the majority of patients being older, virtually all of them are on combinations of drugs. And so to really understand how their networks, how their physiologies are being understood, we can examine kind of the other side, not from the combination, from the patient-centric. There are multiple drugs, almost always affecting their systems. And I wanted to hear any comments you might have on that and how we get to understanding it from the individual patient kind of standpoint of the conference.
Yeah, no, I think that's right on. And I think that by and large, most of the research in combinatorial therapies and trying to hit multiple places in a network happens in oncology, happens in cancer. But actually the vast majority of sort of naturalistic experiments happens in geriatrics, where you're absolutely right. There's, everybody over a certain age has a combination of drugs in their system. Now, those drugs for the most part, we have found are antagonistic rather than synergistic. So we're looking for synergies in these drugs. And I think that just taking a lot of drugs doesn't necessarily get you synergies. As many old folks will tell you, it's miserable. And you take one to offset the other, and that's actually a step towards combinatorial therapy. You take one that makes you achy, and then you take aspirin or opiates so that it doesn't hurt quite so bad. But I'm hoping we can get to better combinatorial therapies. But I do think that geriatrics is one of the great underappreciated places where we could do sort of naturalistic experiments with this kind of work and find synergistic combinations. And maybe using Jamie-like approaches in the geriatric community, we could discover the synergisms that are actually out there already that we just didn't know about.
Of course, it's a small point to add to that, but it doesn't always have to be combinations of drugs, because we're already designed to have things like transcription factors, et cetera, that integrate responses to many genes and many proteins with one single target. And they have a checkered history in terms of being good or safe or effective drugs, but there are a number of single targets that are designed to integrate and affect systems.
I tried to when I originally wrote this, I was going to say that magic bullets never work, but that's not true. There are some magic bullets, and some of those magic bullets affect the integrating point in the system. But I think one of the things that we've learned about, at least in cancer, and growth and proliferation and movement of cells, is there is no one place where it's all integrated. There's a whole system that integrates that information, and that's going to be true of most signaling. And so I think in cardiovascular disease, in cancer, in diabetes, in mental illness, in these systems diseases, there will not be a single target, period. All right, Jamie. Hurt me.
Awesome, Larry, as usual. I think what's interesting when you think about this, and this sort of depresses me, but there obviously is the dimension of time and localization and other variables, and I wanted to sort of tell a quick story and ask you to think about how we're going to work through this question. So I have this new disease that's called acute dog escapist, which means that my dog gets out of my yard. And I've done some investigation, and I've discovered that it's because my fence has fallen down, and my fence is made out of wood. And I know that that chainsaws cut wood, and I have a chainsaw in my garage, and chainsaws run on gasoline. So I started a drug company to eliminate the gasoline in my garage. So can you explain to me how we're going to, given the level of combinatorial issues, resolve my story and get me a drug that keeps my dog in my yard?
Yeah. I would love it if there were no gasoline in your garage. I think that would have social benefit. But let me take that seriously for a second. And I think the question is explanation. Can we build computational tools that will actually get us to cause? And I don't mean grand cause, in the maelstrom of causes in the world. I mean that contrastive cause. So my dog stays in his yard. Okay, and so what we want to do is not look at everything about your dog in your yard. We want to look at my dog versus your dog. Okay, and see if we can figure out stuff. And there's about the same amount of gasoline in my garage as there is in your garage. So a good causal explanation system wouldn't buy your drug discovery thing and would shoot down your startup. And would instead take a look and say, well, gee, Larry's yard, the fence is 6 feet high and it's in pretty good shape. And the problem in Jamie's yard is the fence has fallen down and he needs to fix his fence. And hopefully wouldn't say, give the dog tranquilizers in addition to fixing the fence, because the combinatorial therapy works much better. The woman with the microphone.
Hi. One piece that's missing from that story is that we all know that dogs don't use chainsaws, right? So the question is, I really like what you've done with analyzing some of the metadata. Can you envision an AI approach to tell us what metadata is missing and that we still need to collect?
Maybe. I want to tamp down the expectation. After pumping them up, I want to tamp down the explanation, the anticipation of what AI is really going to be good for a little bit. So, as someone who has to write these programs, it's hard to say, gee, how am I going to get there? And the reason I like the explanation piece is because I have some data about what people do and what shows up in publications, and I have some ideas about how we could take well-represented knowledge of molecular biology and generate possible explanations. Similarly, with the interestingness stuff, I know a little bit about how to write some functions that kind of roughly approximate what I think is interesting, and I know how to learn from watching other people act to roughly approximate what they do. So I think I know, although don't believe me until you see the implementation, I think I know what it's going to take to get to a sort of judgmental interestingness explainer. I think I can build a program like that. I don't know how to build a program that would say, hey, you know what, you forgot to collect this. And it might be that the interestingness piece or the explanation piece points to ambiguities and says, well, could be this explanation, could be that explanation. I can't tell with what I know now. Could be these 5 different possible explanations. That's kind of the way people work, so I expect that would be roughly the way programs would work. And so then we do the normal thing in science, right? Well, I got 5 specific possibilities. I'm going to try and think of clever ways I can distinguish among them by going and measuring something. And hopefully that would be the way that we, figured out what we should go measure. But I don't have any magic bullets for that.
Okay. Quick observations. First, your comment about the sources of phenotypic data, which are mostly, as you said, billing. That's what is euphemistically called administrative data. But restricting ourselves to the fields that are in billing and the unreliability and biases built into maximal billing is a very serious problem. Complementary to that is the need to capture data that don't fit fields at all, that are in the narrative of doctors' notes and nurses' notes, for which there are tools. For example, there's a tool from University of Michigan called Immerse, which is organized to have synonyms and misspellings and other important variables, but to capture important clinical variables that are only in the narrative notes. Also, with regard to AI, there is a fundamental problem which we find in applications to multiplex biomarker studies, and that is that we have many, many more variables than we usually have subjects, which leads to extreme overfitting, which you implied in your comments.
All right, well, let me take those one at a time. There's a wonderful joke that I heard recently. We tried to teach a class on human phenotyping down at med school this semester, and I learned a lot. And one of the things I learned was in the 19th century, patient records were there to remind the physician who the patient was. I see a lot of people, and they didn't remember who he was. It's like, oh, yeah, Mrs. McGillicuddy, I remember her. In the 20th century, they were there to get paid. So I would write down what belonged in I had a diagnostic group, and if I put the right member of that group in, I got paid $100, and I put the wrong member in, I got $20. And so I learned very rapidly how to fill out the records so that I got paid properly. It's kind of like an invoice. And in the 21st century, it's there for legal purposes. So I got to put the right thing in the record to make sure that when I get sued, that I said that I did the thing that I was supposed to do. So if any of those purposes are the purpose that drives the content of the patient record, then we're not going to be able to use it very well for phenotyping, and we're better off taking stuff off the internet than people keeping track of their own weight and their own health status and stuff like that than we are looking at those patient records. Now, that's one of the things I learned in phenotyping class. Was that it's not quite that bad. We do have those textual reports. And furthermore, we have images. We often have pathologic slides and radiologic images and the like. Those are extremely valuable kinds of descriptions of the status of a human being. And the reading piece that I mentioned in the getting the stuff out of the literature is the same natural language processing technology that uses a lot of the same basic ideas about misspellings and synonyms and the like to try and understand stuff in the biomedical literature. And by the way, we have worse problems. We have Drosophila geneticists who thought it was really clever to name a gene the. Okay, bad idea. Don't do that. Anyway, so getting stuff out of the patient record that way is doable and useful, and we can get better phenotypes by looking at the pathologist report or the radiologist report or the discharge summary often than we get from the fielded stuff. So you're right about that. Now let's see. The last one, what was here? Oh, overfitting. Yeah. Well, so. I think the answer to that is the same answer that human beings use, which is knowledge helps. So we're not tabula rasa. We don't know nothing about the data. It's not just a list of numbers. We already have some beliefs about the way things work. And it's sort of, the Bayesian approach, not necessarily formally. I don't want to get into that catfight. But about things that where the data seems to fit in with what we already believe for a lot of other reasons. I don't need huge amounts of data to continue to believe what I already believe. And if the data is showing me something that's completely opposite to everything I've ever believed, then I need extraordinary data to support extraordinary claims. And some version of that is going to be how we make our way through. And I think that the programs that I talked about that integrate stuff from multiple model organisms and lots of different kinds of experiments about genes and the like will provide a very rich background for trying to make sense of the data that we see in the face of the very real possibility of overfitting or jumping to conclusions or something like that, or at least I hope it will.